Deletions mutation. Deletions mutation are those mutations in which one or more than one nucleotides are deleted from the normal sequence of the gene. These kind of mutations, they usually have severe effect on the protein product. Sometimes the protein which is produced as a result of such type of mutations is detrimental. The amino acid sequence which is produced as a result of this kind of mutations is entirely different which was present in the original amino acid sequence of the protein. In this diagram, we can see that what happens as a result of the deletion of a nucleotide. If we do the comparison between the normal sequence of the gene, this is normal sequence of gene, while this is mutated sequence, a sequence in which a nucleotide has been deleted. If we do the comparison between the nucleotides GG, and here we see actually in the normal sequence there was T thymine. This thymine has been deleted. So as a result, this G and this G has come here. So then this C and this G. So as a result of deletion, we see that there is a change in the sequence of the DNA as a result of thymine. This is the second example of deletion mutation. In this diagram, we can see that this is the normal sequence of DNA, while this is the mutated sequence of the DNA. In this diagram, we can see that G guanine, which was present in the normal sequence of the DNA, has been deleted. Due to this deletion, we can see that now the sequence is A, C, and then T is here. So G nucleotide, which was present in the normal sequence of the DNA, has been deleted. So what are the consequences of this deletion? If we see, or if we do the comparison between the normal sequence of the amino acids and the mutated sequence of the amino acids, we see the original sequence was phenylalanine, alanine, valine, alanine, serine, valine, leucine. But now, due to the deletion of guanine or due to the deletion of a single nucleotide, the sequence of the amino acid is phenylalanine and glutamic acid. So in place of alanine, we see that there is glutamic acid, and then leucine, histidine, proline, and phenylalanine. If we do the comparison between the normal sequence of the amino acids and the amino acid sequence which is produced as a result of deletion of G, they are entirely different from each other. This is the second example of deletion. This is the normal sequence of the gene, and this is the deletion. If we see that this T has been deleted, this T that was present in the normal sequence of the gene, that has been deleted. Now the sequence of the mutated gene is double G, T, C, and here was actually at this point there was a T. That T has been deleted. So as a result, we can see the amino acid sequence in the normal gene and the amino acid sequence of the mutated gene, they are entirely different from each other. How mutations, deleted mutations are represented or written? Usually a range is mentioned between the amino acids, those amino acids that have been deleted. In this example, we can see that there is written P cis28 underscore MET30 del. The meaning of this is that the 3 amino acids which are present between 28 to 30, they have been deleted. Deletion mutations are those mutations in which one or more than one nucleotides is deleted, and as a result the amino acid sequence also disturbed when we do the comparison with the normal sequence of the amino acids.